Dear learner, welcome you all in the course of Immunology BZYET 141. Basically, it is a elective courses for the for BSc General program. Dear learner, my name is Dr. Siaram. I am an associate professor, Discipline of Life Science, School of Sciences, IGNOU. Dear learner, today I want to discuss very important topic that is known as working of the immune system first. In this session, we will discuss various things. Then what is the expected learning outcome of today's session? After completion of this session, you will be able to discuss general organization and inheritance of MHC. After completion of this session, you will be able to describe the structure and function of the various MHC molecules. And you will be able to explain how the MHC peptide complex is formed and where the antigen processing and presentation will take place. And after completion of this session, you will be able to discuss the proteolytic breakdown of antigen protein into peptides. And after finishing of this session, you will be able to explain how peptides are transported from cytoplasm to endoplasmic reticulum, endoplasmic reticulum to endocytic vesicles, and finally display of the antigen peptides along with MHC. Then your T cell will recognize or ultimately kill the pathogen and provoke the immune response. So it is expected learning outcome of today's session. Dear learner, my session is divided into two parts, part first and part second. In the part first, we will discuss the various things related to the MSC. Basically, MSC structures, MSC first, second, and third, which are located on the chromosome number 6, and we will discuss their haplotype, MSC haplotypes. I think you know very well the human leukocyte antigen which are present on the on the cells. They will play a role in the graft rejections. In my part second, we will discuss the antigen processing and presentation. So there are the two pathway are there, endogenous pathway and exogenous pathway. There are the several steps in this pathway, antigen processing and presentation. We will discuss step by step in this session. So in part first, I want to discuss in detail. Here is the major histocompatibility complex. So basically, MSC is the collection of the surface protein, a lot of proteins which are present on the cell surface, which are located on the cell membrane of the nucleated cells. So number of the nucleated cells, they have the collection of the surface protein that is known as the MHC. And it is also known as the HLA. HLA is nothing, it is a human leukocyte antigens which are present on the nucleated cells like B cell, dendritic cells, and macrophages. The T cell cannot distinguish between the self and non-self peptide alone. Your T cell cannot distinguish between the self and non-self peptide. It is a very important. Their HLA or MSC is very important. They will display the antigens, or ultimately your T helper cell and T cytotoxic cell, they will recognize it. It is a self or non-self peptides are there, or antigens are there, which are present inside to the on the surface of the cells. The term histo refer to the tissues, and compatibility means cell living together harmoniously. And MSC play a critical role in identify the antigen between self and non-self body. It is also responsible for to the antigen presentation. So your MSC is also responsible for the antigen processing and presentation. They will process it, or ultimately they will present on the surface, or finally your T cell, they are able to recognize it, or immunological response will take place. So it is a very important phenomena, mechanism are there. They will maintain immune response inside to the body. Another thing I want to discuss, the general organization and inheritance of the MSC. In 1951, George Snell showed that the graft rejection are caused by the molecular complex on the cell surface. That there are the certain molecular complex which are available on the cell surface. They can reject the graft of the tissues. If you transform one tissues to the other, your donor to the receiver tissues, they will reject it. It is due to the some molecular complex which are present on the cell surface. He also showed the rejection of the governed by to the group of the genes present in the special place on the certain chromosomes. So these are the chromosomes are their chromosome number 6. They have the group of the gene, they govern it, or ultimately graft rejection will take place. And in 1980, George D. Snell was awarded the Nobel Prize in Medicine for discovery related to genetically determined feature on the cell surface. There are some very important molecules which are present. It is governed by the some genes that regulate the immune response. The immune response that is regulated by the certain gene, it is the first discovered by the George D. Snell. He awarded Nobel Prize in 1980. The MSC determine whether the transplanted tissues or the donor will histocompatible or histo-incompatible within the recipient tissues. So MSC is very important molecules are there. They will responsible for the rejection of the graft. They are the histocompatible or not histo-incompatible are there. The MSC gene are highly polymorphic. They are the very polymorphic are there, because the distinct allele exist in the different individual. So it is so polymorphic are there, or ultimately they will maintain their different individual. The evolution of the MSC polymorphism play essential role in the species survival. So it will help in the species survival. Their role is very important in the evolution. Ultimately, they will maintain the species survival. MSC are epitope that mediate intracellular recognition and the self and non-self recognitions are there. So these are the very important molecules which are present on the nucleated cells. They will take role in the rejection of the graft. They will involve the immune response. Ultimately, your immune response will take place. A lot of antibodies will forms, cytokines will release, ultimately they will activate, or finally the kill the pathogens. These are the very important molecules are there which are present on the all the nucleated cells. There are the three different classes of the MSC molecules are there. Three different class of the MSC molecule: MSC class first, MSC class second, MSC class third. In MSC class first, you can see in these chromosomes, here is the chromosomes are there. It is a long arm, it is short arm, it is a centromere, here is the telomeres are there. It is telomeres. Here is the some very important part are there, that is known as the HLA. HLA antigens. So they have the specific sites are there. In the site there are the certain genes are there, DP, DM, DQ and DR. There are the certain genes which are involved in the MSC second molecule. Here are the A, B, C, D, E, F. These are the number of the genes which are present in the chromosomes number 6. It is a class first MSC, class second MSC. Here is the certain gene which are involved into the class third MSC molecule. In case of MSC class first, it is a glycoprotein found on the all nucleated cell in the body, shows the process antigenic peptides to the cytotoxic T cells. And MSC second also a glycoproteins that are expressed onto the APCs, basically antigen presenting cells. They will express. In this case the number of the cells are there like dendritic cell, macrophages and B cells. They present in process antigen peptide to the T helper cells. So here is these T cytotoxic cells are there. Another is the T helper cells which are involved into the MSC class second molecule. And third one, it is encoded by the certain genes that is known as the MSC class third, and highly diverse. It is the highly diverse structurally and functionally secreted protein. They include the component of the complement system, tumor necrosis factor, heat shock proteins are there. So these are the three type of the MSC molecule are there: MSC class first, MSC class second, MSC class third. They will involve into the process and presentation of the antigens. They will recognize self or non-self antigens inside to the bodies. Another thing I want to discuss the MSC haplotype. So it is MSC haplotype. It is highly polymorphic. I have already discussed about this. The MSC is a highly polymorphic and allelic are expressed into the codominant manner, expressed separately. Codominant is nothing. You can say the alleles are the genes which are expressed separately. The two alleles at a same locus on the homologous chromosome make individual genotypes. MSC class first and class second gene contain many alleles. So you can see it here is the number of the alleles are there which are present on the chromosomes which are responsible for to the class first and class second MSC molecule. Three different type of MSC molecule are found into the long stretch of the DNA on the chromosome 6. I have already discussed it. Here is the chromosome number 6. They have the particular region that is known as the HLA. They have the different class of the, they will encode it, or ultimately they will locate on the surface of the dendritic cell, macrophage and other cells are there. And the class MSC class first is encoded by you can see it here is the A, B, C, D, E, F and G gene, while the DP, DM and DQ and D are encoded to the class second MSC molecule. So you can say it is a haplotype molecule are there. It is highly polymorphic and allele are expressed into the codominant manner, or you can say the expressed separately of each gene, or ultimately they will combine it, or it will express on the surface of the particular cell like B cell, like dendritic cell, like macrophages are there. Another thing I want to discuss the structure of the MSC. The structure of MSC of the class first and class second, you can see in the slide the structure of MSC class first. You can see the structure here is the cytoplasmic tail. It is a transmembrane engagement, or you can say the plasma membranes are there. Here is the MSC molecule. Basically it is a globular proteins are there. These are the globular proteins alpha 1, alpha 2, alpha 3. Here is the beta 2 microglobulin are there. So basically it is a globular structures are there that is made up with the proteins. They have the peptide binding cleft. between alpha 1 and alpha 2. Here is the peptide binding cleft, here is the peptide will bind. I will discuss in detail how it bind. The MSC class first is composed of the alpha and beta polypeptide chains. So you can see it here is the alpha and beta polypeptide chains held together with the non-covalent bond with the short cytoplasmic tails. In the alpha chain, 45 kilodalton, here is the 45 kilodaltons, and is the MSC encoded integral membrane glycoprotein, while beta 2 microglobulin is 12 kd. So here is the 45 kd, here is the beta 2 microglobulin, it is a 12 kd. Is a non-MSC encoded extracellular peptide. The alpha chain is 350 amino acid with their globular domain alpha 1, alpha 2 and alpha 3. So these are the structure of the MSC class first. It has the 26 hydrophobic amino acids, transmembranes. A segment is spanning the plasma membrane. So you can see it here is the plasma membranes are there. The spanning of the plasma membrane, they have the 26 hydrophobic amino acid. So here is the 26 hydrophobic amino acid which are present in this region, and the alpha 1 and alpha 2 form a platform to the peptide binding cleft. Here is the alpha 1, alpha 2, they form the peptide binding cleft, mainly comprising the beta sheet. So basically it is a beta sheet are there. The groups formed between the block or to the both end at to the enough up to the whole 8 to 10 amino acid. So they will hold 8 to 10 amino acid only. So you can see in this group that is known as the peptide binding cleft, they will hold only 8 to 10 amino acid. And beta 2 microglobulin chain is non-polymorphic, and 90 amino acid peptides is encoded by the chromosome number 15 and interact non-covalent with alpha 3, and this part of MSC molecule bind to the cytotoxic T cells. So these are the structure of the MSC molecules are there. MSC first molecule. Another is the MSC second structure of the MSC second molecules. So it is another very interesting molecules. You can see here it is a plasma membrane. They have the alpha 1, alpha 2, beta 1 and beta 2 molecules. Here is the peptide binding cleft. Like MSC first, the class second MSC molecule have extracellular domain and transmembrane domain and intracellular carboxy terminal tail are there. The extracellular domain is a composed of the two non-covalently associated alpha 1, alpha 2 and beta 1 and beta 2. It is non-covalently associated molecules are there of the 90 amino acid anchored to the cell membrane. So you can see the structure of the MSC class second. MSC class second, they have the heavy chains alpha 1 and alpha 1 and alpha 2. These are the heavy chains are there. You can see in these structures has molecular weight of 30 to 34 kDa. The light chain of the beta 1, so here is you can see here is the heavy chains are there, alpha 1 and alpha 2, and beta 1 and beta 2 it is a light chain, has molecular weight 26 to 29 kDa. Here is the 30 to 34 kDa. The transmembrane domain is composed of the 25 hydrophobic amino acid, and the cytoplasmic torque of 10 to 15 hydrophilic amino acids are there. So you can see it here is the 10 to 15 hydrophobic amino acids are there. 13 to 80 amino acid residue in antigenic peptide bind to the antigenic peptide binding domain. So here is in their binding of the cleft, here is 13 to 18 amino acids which are present, which they are able to accommodate in the peptide binding cleft. So these are the very important molecules which are available on the basically nucleated cells are there. It is a dendritic cell, macrophages, and B cells. Another thing I want to discuss the peptide binding groups of the MSC first and second molecules. So you can see here is the binding groups are there of MSC first molecules. Here is the MSC second molecule. Here is the 8 to 10 amino acid longs, but here is the 13 to 18 amino acids longs. They can bound with your antigenic peptide. They will bind to the binding groups are there. You can see here is the amino acid residues. Here is the backbone of the peptide backbone are there. They will bind it. Your antigen will display in the particular antigenic peptides binding clefts are there. The peptide binds on MSC class first group are usually 8 to 10 amino acids, and the residue add to the peptides N terminal and C terminal end to anchor to the groups. Peptide binding of the MSC class second group are usual, it around at 13 to 18 amino acid, and the residue bind to the both end as well as in the middle. But in this case here is the in middle, they will not bind to the binding groups. But in case of MSC molecule, they will bind in the middle also. So it is a peptide binding groups of MSC class first and MSC class second. Another thing I want to discuss the structure of the peptide binding cleft. So you can see the structures of the peptide binding clefts are there. Here is the MSC first, MSC second are there. You can see it. There are the various alpha helix and beta sheet are there. So here is the 8 beta sheet which are present, and 2 alpha helix are present in the MSC class first. You can see the structure. Here is the peptide binding clefts which are available on the it is a part of the MSC are there. So MSC class first and second molecule fold into the highly similar conformation feature to peptide binding groups to the present T cell epitope. They will bind basically the T helper cells and T cytotoxic cells. They act as epitopes. They have the particular peptides, the antigenic peptides are there, or ultimately your T helper cells and T cytotoxic cells, they will recognize it. The peptide binding groups of the MSC class first and class first molecule are composed of the 2 helix. You can see here the 2 alpha helix are there. You can see into the green color. Here is the red color are there. The 2 alpha helix and 8 beta sheet are there. You can see the structure of the beta sheet are there, formed by 1 heavy chains. In class second MSC use 2 domain formed to the different chain to construct the peptide binding groups, and peptide binds or to the MSC molecule through the primary and secondary anchored residue protruding end of the pocket of the peptide binding groups. So basically it is a peptide binding groups or peptide binding clefts are there. Here the peptide will display. Peptide here is the N terminal are there, here is the C terminal. You can see it. Their size it should be it would be 8 to 10 amino acid. Here is the 13 to 18 amino acids are there. They will bind or it will display on the surface of the MSC, or it will recognize by the T cells, or ultimately immune response will take place. Another thing I want to discuss related to the peptide binding cleft or peptide binding groups are there. Peptide preference depend on the amino acid polymorphism comprises to the anchored pockets, and which are related to the various allele of the MSC. So you can see it here is the preference are there. Peptide preference depend on the amino acid polymorphism are there. How many amino acids are present and their different groups are there. Their polymorphism is very important and comprising to anchored pocket which are related to the various allele of the MSC. The conformation of the peptides present on the MSC class first molecule are length dependent. So you can see it here is it is a length dependent are there 8 to 10 amino acid. And the modified group of the post translational modified peptides have essential role in peptide MSC interactions. So their role is very important. Ultimately they will recognize by the T helper cell or T cytotoxic cell, or ultimately they are able to kill the pathogens or and provoke the immune response also. It is another very important things I want to discuss, MSC and antigen presentation. I have already discussed it, the MSC class first and MSC class second are there. So you can see it. Here is the helper cytotoxic cells. Yes, T helper cells are there. You can say here is the CD4 positive cells. Here is the CD8 cytotoxic CD8 positive T cells are there. It is a dendritic cells. They are able to process and present the antigens. Here is the antigen are present on the surface of the MSC. MSC you can see in the red color, or ultimately they will recognize by to the helper CD4 positive T cell. Here is the cytosolic CD4 CD8 cells are there. So it will recognize it, or ultimately they will produce the cytokines, or it will kill the pathogens, or ultimately they will provoke the immune response, or it will generate a lot of cells are there. You can see the MSC and antigen presentation. Antigen processing is a metabolic process. So basically it is a metabolic process. They involve the several reaction in the cells in which the protein are broken into peptides. If any antigens they enter inside to the body, they will broken into peptides, and that can be displayed on the cell membrane together with MSC class first and class second. So you can see here is the antigens are there, or you can say the peptides, broken peptides are there, which are displayed by the MSC class first and MSC class second. It is recognized by T cells, CD8 T cells, and responsible for to the recognition of the antigen recognize of the antigen presented to the MSC class first molecule. And CD4 positive T helper cells, you can see here, cell are responsible for recognizing of the antigen presented with the MSC class first molecule. So you have learned about to the MSC class first and MSC class second. So these are the two very important molecules are there, which are displayed in the dendritic cell, B cell, and macrophages are there. Dear learner, I want to summarize my part first. You have learned the various things related to the MSC which are present in the nucleated cell of the humans, not only human, number of the vertebrates also. And the MSC is known as the human leukocyte antigen complex. You have also learned the MSC gene encoded cell surface antigen presenting protein and of the three classes, MSC class first, MSC class second, MSC class third. The antigen processing enzyme break down to the protein into peptides, and T cell may recognize by displaying them onto the cell membrane using MSC class first and class second molecule. Dear learner, I want to take a short break. Don't go away. We will come within 2 to 3 minutes. Thank you.
Indira Gandhi National Open University, accredited with A++ grade by NAC, provides you a quality education in open and distance learning. School of Performing and Visual Art is offering a wide range of programs in performing and visual art. Certificate program in painting, certificate program in applied art, certificate program in Bharatanatyam dance, certificate program in Hindustani music, Carnatic music and theatre arts.
बहुत यत्न करने के बाद भी नहीं पा सका। संजोग से मैं इस बार लेके आया हूँ।
All the certificate programs are of 6 months duration with 16 credits. We offer 1-year diploma in theatre arts with 10+2 eligibility criteria of 32 credits. The learners are exposed to various aspects of elements of drama, theatre games, acting techniques, scene work procedures, and they will also have the opportunity to take part in a classroom play production. We also offer bachelor's degree in Hindustani music. This program is duration of 3 years and learners are having the flexibility to complete this program over a period of 6 years. Apart from bachelor's, we also offer masters in drawing and painting with a duration of 2 years. Apart from certificate programs, degree, masters, we also offer PhD programs in performing and visual art with a specialization of visual arts, music, dance and theatre. Art is the perfect manifestation of every human being. So what you are waiting for? For online admissions, please visit our website www.ignou.ac.in. For more details, you can also contact Director, School of Performing and Visual Art, Block number 15G, IGNOU, Maidan Gadi, New Delhi. You can also reach us at sopva@ignou.ac.in. Happy learning!
आश्रम के चूहे पकड़े। तुम तो सचमुच राजा लगने लगे।
पता नहीं ये राजयोग कब तक रहेगा।
तो हमारी माया के लिए सुरक्षित है।
Dear learner, welcome in my next part 2. In this part, I want to discuss the antigen processing and presentation by endogenous pathway. So basically, endogenous pathway, the pathogen which are present inside to the body, inside to the cells. You can see it. Here is the antigens are there, the viral proteins are there. They will enter inside to the cells. Here is the cellular protein intact are there. It will move or ultimately they will display it. In the endogenous pathway, the endogenous antigen produced within the cells. The endogenous antigen which are produced and presented, cell are processed and presented. For example, tumor cells which are present inside to the body, mycobacterium tuberculosis which are engulfed by the macrophages, and virally infected cells. And number of these cells are there, they are virally infected. So you can say it is a endogenous pathway. They will process and presented by the endogenous pathway. The endogenous antigen is processed and displayed on the cell membrane with an MHC class first molecule. with the CD8 positive cytotoxic cells identify and degrade it. So finally here is displayed, or it will degrade the pathogens. There are the three steps are involved in the endogenous pathway. So I want to discuss one by one. The first step is antigen degradation into the peptides by the proteolytic cleavage. The various proteolytic cleavage which are involved in this step, they will degrade it, or ultimately it will convert into the peptides. So here is the antigens are there, you can see it. Here is the N terminal and C terminals are there. Here is the proteolytic enzymatic subunit. Here is the 20S proteasome, you can say. It is a proteasomes. They have the proteolytic activity, or they will move inside, or ultimately they will release the peptides. You can see it here is the it is a ATP dependent reactions are there. So you can see it here is the peptides are there. They have the C terminal and N terminal are there, C terminal. Here is the ubiquitin enzymes complex plus ubiquitins. They require the ATP, or it will ubiquitin is bind with their N terminal, or it will recognize by the 26 proteasomes, or it will release the peptides. So these are the structures how the peptides will degrade. The endogenous antigen protein are large and can't bind to the MSC class first molecule, so it required change into the peptides like 8 to 10 amino acid. It need to degrade it by the proteasomes. These are the proteasomes are there. Basically it is a protein molecules are there. It is a group of the proteins, or it will digest, or it will degrade your larger antigens into the smaller peptides. Larger peptide, basically proteins are there, it will degrade it into the peptides. These are small peptides are there, 10 to 8 amino acid by the proteasome. The cytosolic proteolytic system is the cell known as the proteasomes. You can see it here is the proteasomes are there. Here is the 19S regulatory component which are present on this edges of these proteasomes. The proteasomes large component 20S is made up of the 14 subunit. There are the 14 subunit molecules are there. They are joined together. Ultimately they form the 20S proteasomes. And these subunit are arranged in a systematical ring structure. You can see here is the barrel like structures are there. In this barrel your proteins they will pass, or ultimately it will degraded into the peptides. These peptides again further process it, or it will display on the surface of the particular cells along with the MSC molecules. The proteasome consists of the narrow channels. These are the narrow channels are there at each end. These are the narrow channel of the each end which are protein enter. A small protein called ubiquitin is attached. You can see it here is the ubiquitin. It is attached where the target or proteolysis will take place. The ubiquitin protein complex made up to the 20S proteasomes and 90S regulatory. Here is a very important part you can see in the yellow color. It is the regulatory component along with the other things. The process is a ATP dependent. Without ATP this system will not work. Degradation of the ubiquitin protein complex occur in proteasomes and central core and release the peptides. So these are the mechanism how your antigens basically proteins are there. It will degrade into smaller molecules like 8 to 10 molecule of the amino acid, or you can say the peptides are there. This peptide again further process it, or it will display on the surface of the cells along with the MSC molecules. Another step involve in the endogenous pathway, the peptide transport from the cytoplasm to endoplasmic reticulum. So it is another very important process which are involved in the endogenous pathway of the antigen processing and presentations. The peptide produced by the proteasomes in the cytoplasm and transported to the lumen of the endoplasmic reticulum by tap transfer protein. So basically this is the pass through, or it will go to the endoplasmic reticulum by tap transfer proteins, transfer associated with the antigen processing protein, and this procedure necessitates ATP hydrolysis. TAP is a two proteins. You can see the structure of the TAP. TAP first and TAP are there. It is the cytosols. It is a endoplasmic reticulum lumens are there. So these are the very important molecules. The transporter associated with the antigenic process are there. So you can see it here is the peptides. It will pass through the endoplasmic reticulum lumens, or it will go through, or it will process it. This procedure necessitates the ATP synthesis and ATP hydrolysis, and TAP is two protein membrane spanning protein that consist of to the TAP 1 and TAP 2. So it is a very important step. Here is the peptide transport from cytoplasm to the endoplasmic reticulums. So your proteins are there, it will process it, or ultimately it will pass through the TAP 1 and TAP 2. It is a ATP dependent process are there. Your peptides they will enter into the endoplasmic reticulums, lumens. The TAP secrete the peptides, and which the TAP secrete the peptide with 8 to 16 amino acid. 8 to 16 amino acid in length, as well as peptide with to the hydrophobic or basic carboxyl terminal amino acids. A basic carboxy terminal amino acid which are selected for to the use of anchored residues of the MSC class first molecule. So it will pass through the cytosol to the endoplasmic reticulum lumens, or it will basically it is 18 to 16 amino acid in length, as well as peptide with the hydrophobic or basic carboxy terminal which are selected for use of anchored residues of MSC class first molecule. However, optimal peptide length require binding of the MSC molecule, first molecule, and trimming and peptide occur by to the help of to the aminopeptidase in the RER, endoplasmic reticulum. Example ERPD. Here is endoplasmic reticulum endopeptidase, aminopeptidases are there. So these are the very important enzymes are there. They will help in trimming of your peptides. So it will make 8 to 10 amino acid. Therefore, TAP deficiency can lead to the autoimmune diseases. If any person they deficient with the TAP, so you can say this person is suffering from the autoimmune diseases are there. And third step which are involved into the endogenous pathway, that is known as the peptide assembly formation of to the MSC class first molecule. Here is endoplasmic reticulum is responsible for production of the alpha chain and beta 2 microglobulin of the MSC class first available into the cytoplasm. So here is endoplasmic reticulum is they have the very important role. They will produce the alpha 1, alpha 2 and alpha 3 and beta 2 microglobulin molecules of MSC which are available in the cytoplasm. For the formation of MSC molecule, first it need to ensure the endoplasmic reticulum and block the peptide binding cleft up to the MSC. So here is a very important part that is known as the peptide binding cleft which are present in the MSC molecule. It need to the block it, otherwise your MSC will not work. So it need to block the peptide binding cleft. And there are molecular chaperones are involved in this process. Selexin, it is the first chaperone or molecular chaperones are there, is involved to the making of the assembly of the MSC class first molecule. And selexin is an endoplasmic reticulum resident membrane proteins. So it is found into the endoplasmic reticulum protein that support to folding of the free class first chain. And selexin is released when the MSC first molecule bind to the beta 2 microglobulin. When the alpha and beta, these are the two molecules, they will bind together. So ultimately selexin they will remove from this place. An MSC first molecule associated with third and fourth chaperone, that is known as the trypsin and calreticulin. So these are the two another chaperone molecules which are involved in the process of the peptides formations in case of the MSC class first expression. Another thing I want to discuss, the MSC class first peptide loading complex. It is another very important mechanisms are there. It is first given by the Hammer et al. In 2007. You can see it here is the peptide pool are there. There are the trypsin. I have already discussed. It is a chaperone molecules which are available in the endoplasmic reticulums. Another is very important molecules that is known as the ERP57s or CRT. These are the very important proteins which involve into the expression of the MSC or your peptide. They will bind together. Another very important molecules, you can say it is a type of the enzymes that is known as the endoreticulum trans aminopeptidases are there. It will trim it, or ultimately it is your mature MSC molecules. They have the antigenic peptides are there, or it will recognize the CD4 CD8 positive cells are there. So these are the trypsin enable to acquisition of endogenous antigenic peptide, bringing to the bearing peptide into close proximity with MSC class first. So these are the very important mechanisms are there of loading complex of the peptides on the MSC class first. So these are the mechanism, or ultimately your peptide molecule they will load on the peptide binding cleft of the MSC. Another mechanism that is known as the antigen processing and presentation by the exogenous pathway. Exogenous antigen is produced outside of the cell, bacteria, virus, and intracellular pathogens. So these are the outside of the cells, like bacteria, like fungi, like virus, which are available outside of the cells are there, are outside of the body. They will infect it, or ultimately they will display on the surface of the MSC second. Firstly, the antigen is phagocytized by the antigen presenting cells. The antigen presenting cells, it may be B cells, and dendritic cell or macrophage cells are there. The APC, antigen presenting cell, internalize the antigens. Antigens, it may be bacteria, it may be fungi, or it may be virus, which are available on the outside of the cells. And antigen are phagocytized in endocytosis. The other APCs are either non-phagocytized or poorly phagocytized. For example, antigen is internalized by the B cell receptor mediated endocytosis, and it is a non-phagocytic. And finally, exogenous antigen is displaced onto the cell membrane within the MSC class second molecule. It is recognized and degraded by to the CD4 T helper cells. Another thing I want to discuss, there are the antigen presenting cells, and some antigen presenting cells which are the professional antigen presenting cell and non-professional antigen presenting cells which are available inside to the body. So there are the professional antigen presenting cell, that is known as the dendritic cell, macrophages and B cell. Dendritic cell, primary function of the dendritic cell, the capture, process, display antigen on the cell surface. And macrophages are vast and specialized cell capable of the recognizing, engulfing and displaying antigen on the cell surface. And third one is the B cells. B cell also express the major histocompatibility complex MSC first and second, and are equipped with all the machinery required for to the antigen uptake, processing and presentation. So there are the three cells are there which are the professional antigen presenting cells are there. They take very important role to provoke the immune response inside to the body. Another is the non-professional antigen presenting cells are there. The fibroblast and glial cells in the brain and pancreatic beta cells. So these are the three very important cells are there which are non-professional antigen presenting cell. The under physiological condition, in case of the fibroblast, the under physiological condition antigen presenting fibroblast subset have been identified in the human lung, liver and colon by expression of the MSC second molecule. And another is the glial cells in the brain, microglia represent to the main APCs inside to the brain parenchyma during the neurodegenerations. And third one is the pancreatic beta cell. It is also involved into the antigen presenting cell in the pancreas. Unfortunately, the immune system numerously kill the beta cells in the peoples with type 1 diabetes as a result of the pancreas cannot produce insulin without the beta cell. So these are the non-professional antigen presenting cells are there. I want to discuss the exogenous pathway of the antigen process and presentations. In this case, there are the three steps which are involved in the antigen processing and presentations. First one is the endocytic vesicle peptides are made from the internalized the antigens. In this three acidic compartment are involved. In this pathway, first compartment, you can see it here is the early endosomes are there. Here is the antigens. Antigens they will endocytosis or it will take place. It will fuse with the endosomes. Here is the their pH at around 6.5. 6 to 6.5. It is a first compartment. Another is the second compartment, and third one is the here is the third compartments are there. In first compartment, early endosome, you can say the early endosome, the pH 6 to 6.5. In second compartment, later endosome, their pH 5 to 6. And third one is the lysozymes, pH 4.5 to 5. To internalize the exogenous antigen transported from the compartment up to the next encountering. So there is the early and late and in the their endosomes are there. They will convert into the peptides from one encountering the hydrolytic enzymes and decreasing the pH. An antigen is broken down into 13 to 18 amino acid peptides are there. So here is required 13 to 18 amino acid, but in case of endogenous, it require 8 to 10 amino acids. So it will pass through the early endosome and late endosomes and lysosomes are there. So ultimately it will display on the surface of the cell surface along with the MSC class second molecules. So it is a first step. Another is the second step, transport of the MSC class first molecule to endocytic vesicles. So your antigen, they will transfer to the endocytic vesicles are there. The endoplasmic reticulum, where the MSC class second molecule are made, the peptide binding cleft of the MSC class molecule is blocked by to the trimer protein called the invariant chain CD74. So these are the very important molecules, or it will maintain the cleft. The invariant chain prevents CD74 cellular endogenous peptide in attaching to the cleft. In addition, the invariant chain cytoplasmic tail contain the sorting signal help in MSC class second molecule migrate from endoplasmic reticulum to endocytic vesicle. So it is another very important step which are involved into the exogenous processing and presentation of the antigens are there. And third one is the third one is the peptide assembly bind transported MSC's class second molecules. So you can see the figures are there. Here is the they will transport it. The peptide assembly will form and transport to the MSC class second molecule. The proteolytic activity increase into the endocytic vesicles and invariant chain and slowly dissolve and release to the just little piece known as the clip. The clip here is the CLIP class second associated invariant peptides are there and clips. Here is the proteolytic activity increases in the endocytic vesicle and invariant chain slowly dissolved and releasing just little piece known as the CLIP. And CLIP bind to antigenic peptides and peptide binding cleft of the MSC second molecule prevent them binding prematurely. So ultimately your peptide binding cleft they are safe. After that, the non-classical MSC class second molecule known as the HLA-BM remove clip CLIP from the endosomes and substitute it the exogenous peptide 13 to 18 amino acid, and the stable MSC second molecule with antigenic peptides is then presented into cell surface, and T helper cell will recognize the complex and trigger to the suitable immune response, take secretion cytokines and chemokines to control the infections. Dear learner, I want to summarize my part second. I hope you have learned the number of the things related to the MSC and antigen presenting and processing. Here is CD8 positive cell recognize antigen presented along with the MSC class first molecule, and antigen delivered along with to the MSC class second molecule is recognized by to the CD4 positive T helper cell. There are the two type of antigen processing and presenting pathway. So you have learned the endogenous pathway, exogenous pathway. In the endogenous pathway, the endogenous antigen generated within the cell and processed and presented. In MSC class first molecule used to deliver the processed endogenous antigen onto the cell membrane, which is recognized and destroyed by to the CD8 positive T cells. You have also learned the specialized antigen presenting cells used to exogenous pathway to present and peptide produced from the proteins that is cell are endocytose, and MSC class second molecule are used to the present the peptides. I hope you have learned number of the things related to the MSC first, MSC second, and antigen processing and presentations, how to antigen present and process it. Dear learner, if you want to study detail, you go through the IGNOU SLM BZYET 141 unit number 9. You can go through the literature, or you can clarify each and everything related to the MSC and antigen presentation and process. Thank you.